This is your favorite professor, Professor Scam, clocking in to talk to you about mutations. So, okay, I'm being really technologically inept today, so bear with me. Okay, there we go. All right, so what is a mutation exactly? Well, often mutations are referred to as an alteration in DNA sequence, or the changing of the structure of a gene, resulting in a variant form. And if it persists through cell division, in other words, if it's not repaired, that mutation then becomes quote unquote fixed in the genome, right? You have something that persists through cell division. And when it persists through cell division, becomes fixed, and that means it can be then transmitted to subsequent generations. If your mutation is also fixed in the genome, that's when you can actually find those mutagenic differences. So you can take two sequences and compare them and find that actual difference in the sequence. Now these differences, all right, what are they caused by? Well, they can be caused by a number of events. The alteration of single base units in DNA, or the deletion, insertion, or major alteration to the structure of the chromosome itself. So, for example, you have a translocation event, right? And that can change the length of the chromosome. By changing the length of the chromosome, you can then cause cell death. Now, the cellular location of the mutation is very important. So, eukaryotic organisms, they have two primary cell types. You have your germ or your germline cells and your somatic cells. Now, mutations can occur in either cell type. So, if a gene is altered in a germ cell, that means it's either in your egg or sperm, the mutation is termed as a germline mutation, right? Because it's in your germline cells. Your germline cells or your germ cells are also called your myocytes. Now, because your germ cells give rise to gametes and occur in your sex cells, these mutations can be transmitted to a succession of generations. Some gametes will carry the mutation, and it will be passed on to the offspring. And if it's inherited, all the cells of the offspring will be affected. Your somatic cells, on the other hand, these give rise to your non-germline tissues. Right? So these are your guys over here. So these are your non-germline tissues, or what we call your body cells. So these are the cells that you find in your liver, your stomach, intestinal lining, etc. Now, when you have a mutation that occurs in the somatic cell, it's called a somatic mutation, right? Hence the name. So these mutations are often referred to as acquired mutations. It does not come from a parent, but rather is acquired sometime later. And it starts in one cell, and then from that one cell, it can be passed on to any new cells that are created from that one cell. So it can be kind of spread through cells via replication events. However, since your somatic cells are not involved in gamete production, these mutations can't be passed on to your offspring. So they're not inheritable. And most of your cancers are caused by these somatic mutations, or quote-unquote acquired mutations. For example, your melanomas or skin cancers, lung cancers, they're somatic mutations. Does that mean all cancers are somatic mutations? No, absolutely not. So there are some cancers that are germline mutations rather than somatic. Most of your family cancer syndromes are caused by inherited mutations, usually in a tumor suppressor gene. Now, your tumor suppressor genes, these are genes that normally keep cells under control by slowing down how often they divide. I like to think of them kind of as the bouncers at a club. They are like, all right, guys, like, keep it under control, calm down. Like, they keep a handle on everything. They keep those little genes kind of in line. Don't let them get too rowdy. The The tumor suppressor genes also help in repairing any mistakes in the DNA. They also play a role in programmed cell death. So they help to tell cells to die at the right time. Now that's when these guys don't really seem like bouncers. Bouncers don't tell people to drop dead. I'm sure they want to, but they don't. But you get the point, right? Your tumor suppressor genes that keep everything regarding cell growth kind of in control and regulated. Now, when that gene stops working as a tumor suppressor gene, that means cells can start growing out of control. You start having this uncontrolled cell growth, you get the metastases, all of a sudden you have cancer. Uncontrolled cell growth is what leads to cancers. So one example is this syndrome called the hereditary breast and ovarian cancer syndrome, HBOC. So this is when you often see breast cancer and ovarian cancer running together in families. Another is Lynch syndrome. So this results in both colon and endometrial cancers. Again, these are another family cancer syndrome. Von Hippel-Lindau disease is another. So not only is it important to understand the cellular location of the mutation, but it is crucial to understand the rate or frequency at which it's occurring. So this is called your mutation rate, and that's the rate at which it occurs in a population or at a specific locus of the chromosome. Now, by knowing the mutation rate, you can predict the frequency of a mutant allele in a population, or investigate if parts of the genome mutate faster than others. You can also start obtaining information that allow you to determine frequency of mutation according to demographic factors. So these are factors such as ethnicity, age, or gender, which often have correlations with different mutations or the different variants of a mutation. Mutation rate and cell division or replication time are also pretty proportional. So here, the smaller the genome size, the faster the mutation rate. As a result, you have a higher frequency or commonality of mutation. So for example, your viroids, right? So they have the smallest genome size. Their genome only ranges between 10 to the second, 10 to the third power. But they have the fastest mutation rate. On the other hand, or other end of the spectrum, you have your higher eukaryotes, right? We have much larger genome sizes, but much lower mutation rates. And that's also kind of a factor of, again, how quickly we go through cell replication, right? So things that are able to have high cell replication times or replication events, they're going to have faster mutation rates, right? That mutation is going to be replicated faster, or there's a higher chance of the mutation occurring, right? So again, the smaller genome size, the faster their cell replication, meaning the faster their rates of mutation. So when we categorize mutations, there's usually 4 bins that we use to categorize them. There's many ways that you can categorize them. These are kind of the main ones. So mutation can either be spontaneous or induced. Spontaneous are those that occur randomly, meaning they have no obvious or apparent cause or reasoning. Your induced mutations, on the other hand, they're produced by some known factor. So for example, exposure to radiation, especially high gamma or UV radiations, or exposure to harmful chemicals in the environment. They can also be categorized by the location of the mutation. Many are categorized by the actual type of molecular change that's induced. So, for example, was an adenine converted to a guanine, or was an adenine deleted, or was cytosine replaced for thymine, etc. And then you have your phenotypic effects for categorizing, the actual influences to the observed traits of that individual. Now, you often can't put a mutation in a specific little category bin. So you can't just say this mutation, or you can't categorize mutation just by a phenotypic effect. There's a lot of overlap, and you can discuss one mutation in more than one way, often because, well, you change the type of molecular change, you're going to often have some sort of phenotypic effect. So they often go hand in hand together. With that said, there's no right or wrong way, per se, to categorize a mutation. Again, you can categorize it by the type of molecular change. You can categorize it by the combination of type of molecular change, whether it's spontaneous or induced. You might have something that's an induced induced silent mutation, for example. Again, you can't, most of them are overlapping. Oh, computer, there we go. So here we're looking at categorizing by molecular change. That's kind of the universal one. You'll see that in any combination of names, per se, given to different types of mutations. Now, frequently, your mutations result in the substitution of one base, something called a point mutation, right? So your point mutations are pretty frequent, hence why I'm kind of focusing on them here. Now, your point mutation could be, again, it's a substitution of one base. So this can be a change in nucleotides, the removal or inclusion of a base, anything regarding the change of one letter or one nucleotide, one base. So there are 6 kind of fan favorites of point mutations. All of these occur only in the coding sequences. So as such, they could affect protein folding if that substitution involves amino acids of a different polarity, meaning they have a different chemical character to them, right? So if you switch one amino acid that is polar in character to an amino acid that is nonpolar, you're going to influence how that amino acid interacts with adjacent amino acids or neighboring amino acids, which is going to affect the protein folding and therefore influence the 3D structure of that protein, right? You're going to shift where different bonds occur between amino acids and kind of how they're held together spatially. An example, right? Sickle cell anemia, that's a big one. So we were talking about that with protein translation. When you affect the actual sequence of amino acids, that can have these profound effects on the actual structure of the protein and therefore the phenotypic effects are profound. So sickle cell anemia is a great example of point mutation. So this is when you have the substitution of glutamic acid, which is occurring at position 6 in the chromosome. That's being substituted for valine. Now, they're both of different chemical character. Glutamic acid is an acidic, electrically charged amino acid, whereas valine is nonpolar. So that change in the chemical behavior influences and alters how the protein folds. Now, that protein that they're affecting is the beta subunit of hemoglobin. So they cause this mutant form of the beta subunit, which then is going to interact differently with the other beta subunit and the alpha subunits, your alpha 1 and 2 subunits of hemoglobin. So therefore you get this actual mutated form of hemoglobin. And that hemoglobin is going to start interacting differently with other hemoglobins. And then before you know it, you have blood clots, or you have these sickle-shaped hemoglobins that just start piercing through the blood vessels, causing sickle cell crisis or bleeding out. Again, that's all just from one little base substitution. So, alas, I diverse. Back to our little cast of characters here. So, these are the main types of point mutations. I just want to discuss them briefly, somewhat briefly. So, missense mutation. This is a type of mutation where you have a change in one DNA base pair that results in the substitution of one amino acid for another in that protein, whatever protein is made by that gene. So, in other words, you're changing a single nucleotide. That alters the codon, meaning you get a different amino acid that's encoded. Those are missense mutations. Nonsense mutations, I remember them because they're just a bunch of nonsense to me, right? So these are also substitution of one base pair, but instead of substituting an amino acid for another amino acid, you're instead swapping the coding for amino acid to become that codes for a stop codon. So in other words, you're going from, you're substituting an amino acid with a stop codon. So the altered DNA sequence now prematurely signals the cell to stop building the protein, which results in a shortened protein that is either not going to function properly or is not going to function at all. So again, it's a bunch of nonsense. You also lose genetic material because of that premature halt. Silent mutations. So these guys are sneaky. These are like the little ninjas of your mutations. Hence the name. So this is because they have no effect on the encoded protein. So what happens is you have a change of a single base pair, right? And that transforms the codon to a different codon. But that codon is still coding for the same amino acid as the previous. So again, the amino acid that's translated is still the same. And if we think or go back to our codon tables, you can have an amino acid coded for by several different codons, right? You don't have one codon that codes just for one amino acid. This amino acid can be coded for by 3 or 4 different codons. So here you're basically swapping those codons around in that same grouping. So you can have this alteration in the codon, but you're still getting, if you were originally getting an arginine, you're still getting an arginine, even with that difference, or you're still getting serine for serine. Like you're still getting the same amino acid, even though you have a different codon. So again, they don't have an effect on the encoded protein. They can only be found by actually sequencing the DNA and seeing that change or substitution in the base. Transition mutations. So this involves swapping a purine for a purine, right? So swapping adenine for guanine or guanine for adenine, vice versa. Or swapping pyrimidine for pyrimidine. So swapping thymine for cytosine or vice versa. Transversion, on the other hand, is just the opposite. So this involves swapping a purine for a pyrimidine or a pyrimidine for a purine. So this could be swapping an adenine for a cytosine or vice versa, for example. Frame shift mutations. Okay, so these ones are really interesting. So this is a mutation that results in a shift of the entire reading frame. And that's either due to insertion, which is the gain of one or two nucleotides, or deletion. This is the removal or loss of one or two nucleotides. Now, the addition or loss of DNA bases changes a gene's reading frame, right? So a reading frame consists of 3 bases. Those 3 bases create a codon, right? Each codon codes for one amino acid. If you start adding or taking away certain bases, you alter the grouping of those bases. So the frameshift mutation shifts that grouping of the bases. So you then change the triplet pairing, or not triplet pairing, the triplet coding for each codon. Therefore, you get different codons, which is going to change the amino acid coding. So this usually will result in a protein that's not functional. And again, they can be any insertion, deletion, or duplication events. All of those can cause a frameshift mutation. Now, there's one other I don't have up here, but it's called a repeat expansion. So nucleotide repeats are, or I should say, in this type of mutation, you have these nucleotide repeats, right, which are these little short DNA sequences that are repeated a number of times in a row. Your repeat expansion mutation will increase the number of times that short DNA sequence is repeated. And this can, again, result in improper protein functioning, or the protein will not function at all. So when we're categorizing by phenotypic effects, right, these gene mutations, they can have varying effects on health depending on where they occur and whether they alter the function of essential proteins. So here I have a picture of yellow Drosophila melanogaster, right? So this little yellow dude has a different allele than the wild type. So he gets a little lighter of a body color. Interesting is the fact that it actually won't mate as frequently as the wild type. So when we're thinking about how we can actually classify this mutation in this particular instance, is it classified as behavioral, morphological, biochemical? Those are usually how we classify mutations phenotypically. In this case, it can be classified as all of these, right? Morphological is your physical appearance. So there is a morphological change, right? You have the lightening of the body color. Behavioral, it's not mating as much. Biochemical, it has different hormones and chemical properties that actually result in or cause it to not mate as much. Those are all phenotypic effects, and it kind of goes parallel to this idea of pleomorphic genes, which are genes that create a variety, quote unquote, a variety of phenotypes that are seemingly unrelated. So for example, the Goody gene in mice. So when you have a mutation, or the mutant form of the Goody gene, it's a dominant mutation, and it causes yellow fur. Now, these little guys are, with the yellow fur and the mutant form, they're predisposed to adverse health conditions. So the heterozygotes of this mutation will get really fat. They have a lot of cardiac issues. They usually die prematurely. I think they're also prone to diabetes. They're just like doomed from the start. Your homozygotes, you won't even get to see them because your homozygotes have what's called the lethal mutation. So if they're homozygotes for that mutant form of the Goody gene, they will die during embryonic development. So that actually changes the Mendelian genetics of the whole thing. So mutations can arise in a number of ways. Errors during DNA replication, repair, or recombination can lead to base substitutions, insertions, or deletions, as well as mutations that affect longer stretches of DNA rather than just one or a few base pairs. Now, usually they'll occur either from one, DNA damage, that's followed by erroneous repair, which is then followed by cell division. Now, that's important to remember because if you have erroneous repair, that's then followed by cell division, that's what fixes the mutation in the genome. So that's one. The second is errors, or the second is errors during replication that are not corrected. So you kind of need both to go hand in hand. So really, mutations are some changes in DNA that persist after replication, meaning it's not fixed and it's not corrected, and then it'll go through replication and it's still not fixed. So example, DNA polymerase slippage, right? So this is an error that can be introduced during replication and occurs frequently on the lagging strand during DNA replication. It also occurs often in areas where you have repeated or repetitive sequences. Now, when a DNA polymerase is going along your little template strand and it encounters a run of repeated sequences, at this point, it can't effectively clamp onto the template. It's kind of like a little fish out of water. It's not really securely held to the template. So as a result, this causes slippage, and it causes improper replication, resulting in a shorter or longer sequence. Sometimes it will actually skip over some of the sequence in that strand of DNA. So Tautomers, these guys are sneaky. Tautomers are alternative chemical forms of bases that differ by one proton. And the reason I'm bringing them up is because these are the guys that kind of allow for these erroneous base pairings to go unnoticed through DNA replication. Again, they're very sneaky. So with your Tautomers, what's happening is you have each of two or more isomers of a compound that exist together in equilibrium, and they're readily interchanged, meaning they readily kind of swap back and forth with each other by migration of an atom or group within that molecule. And this is all on the basis that your nitrogenous bases will readily shift in form. And that's because they shift their protons due to electron movement. So if your bases, what they'll end up doing is actually shifting the hydrogen bonds. So a proton moves from one location to another. It alters where that hydrogen bond is forming. It'll often move the location of double bonds. So a double bond can move from between a carbon and an oxygen to instead a carbon and a nitrogen, as you see here. So the bases can shift form, which changes how they pair. They also have this really annoying tendency that once they shift to this abnormal form or their isomeric form, they will then shift back to normal. So they go from normal to abnormal, and then from abnormal back to normal. So again, this is all just the natural tendency of nucleotides to change their bases. And you see this in cases of, again, incorrect base pairing, just due to that random tendency. So you're probably sitting there, you might be drooling at this point, not sure, but you're probably thinking, all right, so what? What's the big deal? Well, the key fact is that tautomerism, again, alters the base pairing properties of a nucleotide. Now, since the genetic code is, in fact, the binary code of hydrogen bonding, this means that tautomers intermittently masquerade as other members of the code. So here's the big four, right? You have your thymine, adenine, cytosine, guanine, right? So these are the normal standard base pairing arrangements. Thymine always goes with adenine, right? They're a happy little couple. Cytosine always goes with guanine, another happy little couple. And then we have this crap show occurring down here, right? So what's happening here is you have tautomerization, right? Tautomerization is the result of interactions with water. And that's where you're getting these movements in your hydrogens, or your protons, sorry. So some are moving to a different location or coming from a different location. And that's how the tautomeric forms then lose the ability to make the correct base pairings. And so not only do they lose the ability to make the correct base pairing, so now because of this tautomeric shift, thymine has, and specifically the tautomeric shift, you have the shift, it's almost like a weird resonance structure. So you have the shift from carbon double bonded to an oxygen. You have that shift or that movement in protons. So now your carbon is double bonded with nitrogen instead rather than oxygen. So this alters the hydrogen bonds that then result from, or that then occur or form with that. So here, thymine has gone into its tautomeric form, its weird little isomer form, because of its natural tendency to shift its protons. So it's lost the ability to make the correct base pairing, right? It's no longer base pairing correctly with adenine. Instead, it's making this incorrect pair with guanine. And that's kind of the worst part, right? It's able to create these viable pairings with incorrect partners. So they are able to exhibit unconventional base pairing, which is not cool. I mean, it's really neat that they're able to do that, but not cool when you are talking about mutations. So this is a diagrammatic representation of the nitrogenous bases and their possible tautomeric states. So you have these amino forms, you have ketonic states, and then here you have so these are their normal pairings, and then you have the abnormal pairings. So here this is the regular guanine, right? Here's your abnormal guanine. So this is the isomer. That little star indicates the isomer form. So again, this shows that you now have these improper pairings that are formed, and they will persist. So for example, here we have, so we start off, I want you to focus on this little base pairing here, right? We have thymine base pair to adenine. We go through a little replication event. So this double helix gets separated by helicase. You have your two strands. At this point in life, your little amino acid A, sorry, not amino acid, sorry, your adenine that's up here decides to shift its form. So it does a tautomeric shift into the amino form, right? So now it's no longer your regular amino A, right? Amino A is the normal form, your normal adenine. It is now your abnormal A, the amino form. It's not amino A, it's amino A. So it's done this little shift, and as a result, it's now a tautomer. Well, in this abnormal form, it creates an anomalous base pair with cytosine, right? So that T base, what happens here is you get the synthesis with this template, right? So because you have this abnormal form here, when this new strand is synthesized, you have cytosine placed here rather than thymine, right? So you have this abnormal base pairing due to this improper or abnormal adenine. So you get this double helix. It's going to go through another replication event, right? So the helicase to double helix separates by helicase. At this point, your little naughty tautomer decides it's going to switch back to normal. So it switches from its abnormal form, the amino A, to its normal form, amino A, right? So that's all well and good here. This is great for this strand because now you're going to get your regular thymine base. But not so hot over here, right? Because you're still persisting with the cytosine. This kind of goes its own way, this will go its own way. So because you have a cytosine here, right, this strand is going to be used as the template for the synthesis of the replicate strand or the complementary strand. Because you have a cytosine here, you're going to get a guanine. Okay, that's a correct base pairing, right? But it's not correct in terms of its kind of its homologue. So you get what's called a transition mutation. So if you lined this helix up with this helix, right, you have a transition mutation from guanine, adenine, cytosine, and thymine. So that's what I'm referring to as these tautomers, the naughty tautomers, can go from normal to abnormal, and then from abnormal back to normal. And that's going to affect this second strand that's replicated. All right, so I think what occurs next is DNA repair systems. So I'm going to end there with mutations. You can tune into the next video for DNA repair systems. Otherwise, have a good evening.